Let me ask you something. Have you ever wondered why your body knows what to do every second? Like how it grows hairs or how it heals from wounds. And have you ever wondered why you look like your parents? Behind the scenes, your body is constantly reading and copying genetic instructions. These instructions are written in a molecular language made out of chemical letters, and those letters are purines and pyrimidines. They're not just tiny molecules, they are the foundation of your genetic code. Today, you are going to learn how to read this alphabet of life, how to tell these molecules apart, and how they power everything from inheritance to protein synthesis. Before we dive in, let's play a quick game to get you thinking. I'll flash a few names and your job is to decide if it's a nitrogenous base or it's totally made up.
So now we are going to have our minigame, real or fake base.
Identify if the following items are real or fake. Feel free to pause the video if you need more time.
For our first item, guanine. If you answered real, you're correct. Guanine is a purine. Kryptonine. If you answered real, better luck next time. Kryptonine is fake. Cytosine. If you answered real, congratulations, you are correct. Cytosine is a pyrimidine. Blastidine. If you answered fake, you are correct. And for our last item, we have Uracil. If you answered real, congratulations. Uracil is an RNA-only pyrimidine.
How did you do? If you already knew some, great. If not, that's okay. You'll get to know them all by the end. To start, here are the learning outcomes.
Here's what you'll master by the end of the session. Pause and take time to read these so you will be guided all throughout.
To start with the basics, what are nitrogenous bases?
Nitrogenous bases are nitrogen-containing molecules found in DNA and RNA. They are part of each nucleotide, which includes a nitrogenous base, a sugar, and a phosphate group.
Think of bases as a letter, and the other parts is the frame.
Checkpoint. Identification. What are the three parts of a nucleotide? If you answered nitrogenous base, pentose sugar, and phosphate, congratulations, you are correct.
Now let's move on to the structure and comparison. Purines and pyrimidines are classified based on their chemical structures.
Purines and pyrimidines are classified based on their chemical structures. Purines, like adenine and guanine, have two fused rings, a 6-membered and a 5-membered ring. That's why they're larger. While pyrimidines, like cytosine, thymine, and uracil, only have one ring, making them smaller.
Quick guide. Cytosine and adenine has amino group, while guanine, cytosine, thymine, and uracil has carbonyl group, while methyl has polythymine.
The bond between the sugar and the base occurs at the nitrogen atoms. Nitrogen 9 for purines and nitrogen 1 for pyrimidines.
This structural differences is essential for understanding base pairing and maintaining DNA's consistent width. Mnemonic time! For our first mnemonic, we have pure as gold, where adenine and guanine are purines. And for our second mnemonics, we have cut the pie, where cysteine, uracil, and thymine are pyrimidines.
Here's another checkpoint to check out the things that you have learned. This checkpoint is true or false. For our first item, we have thymine is a purine. If you answered false, you are correct. For the second item, purines are larger than pyrimidines. If you answered true, you are correct.
Let's proceed to DNA and RNA. Who has what?
Now let's talk about its molecule and the bases found. For DNA, we have adenine, guanine, cytosine, and thymine. While for RNA, we have adenine, guanine, cytosine, and uracil.
Both DNA and RNA use 4 nitrogenous bases, but they don't use the exact same ones. RNA swaps thymine for uracil.
Why? Because RNA is a single-stranded and uracil is more stable and energetically favorable in that form. This one-letter change is critical in transcription. Time for another checkpoint. This checkpoint contains multiple choice question. For our question, which base is found only in RNA? A. Cytosine, B. Guanine, C. Uracil, and D. Thymine. If you answered C, uracil, you are correct.
Now, the tricky part. Base pairing rules. The rulebook for how DNA strands bind.
Adenine pairs with thymine via 2 hydrogen bonds. Guanine pairs with cytosine via 3 hydrogen bonds. In RNA, adenine pairs with uracil instead of thymine. One purine always pairs with one pyrimidine, ensuring the double helix stays uniform. More bonds like guanine and cytosine mean a more stable region of the molecule.
It is important during processes like replication and denaturation.
Checkpoint. This checkpoint contains multiple choice question. For the question, which base pair has 3 hydrogen bonds? A. Adenine and thymine, B. Adenine and uracil, C. Guanine and cytosine, D. Thymine and uracil. If you answered C, guanine and cytosine, you are correct.
Now that we know most of it, let's talk about why it all matters. These bases are just static letters. They are:
first is storage units for your genetic information. Second is instruction codes for making proteins. And third is templates for DNA replication.
That's why accuracy in base pairing is important.
Checkpoint. True or false? Base pairing ensures DNA is copied accurately. If you answered true, you are correct. Uracil pairs with guanine. If you answered false, you are correct.
We're almost there. Apply your knowledge in real-time practice. Let's do a quick practice to help everything stick.
Sample DNA strand 5' 𝐂𝐬𝐧𝐢𝐴 3'.
First, let's write the complementary DNA strand. Remember, A pairs with T, T pairs with A, G pairs with Z, and C pairs with G.
So the matching strand is 3', TACGAT 5'.
Now, let's write the mRNA transcript.
This time, use the same pairing rules, but uracil replaces thymine because RNA doesn't have T or thymine. So adenine is to uracil, thymine is to adenine, guanine is to cytosine, and cytosine is to guanine. So the mRNA will be 5' 𝐂𝐬𝐧𝐢𝐴 3'. This is how your body uses DNA to make mRNA. This process is called transcription.
It is basically how your genetic instructions get read and carried out.
This is exactly how we apply base pairing knowledge in biochemistry, genetic diagnostics, and even drug development.
Let's bust some common myth.
Myth versus reality. Thymine is found in RNA. Reality, thymine is only found in DNA. Myth, uracil is a purine. Reality, uracil is a pyrimidine. Myth, guanine pairs with adenine. Reality, guanine only pairs with cytosine. Myth, all bases are in both. Reality, thymine and uracil are exclusive. It's time for another checkpoint. This checkpoint is identification. Which base is only in RNA and is a pyrimidine? If you answered Uracil, you are correct. Now, for this segment, we will conduct a fast-paced 1-minute game called Base Match.
Nice work! You have now understand the chemical alphabet of life and how it shapes who you are. Congratulations, you have now cracked the code of science the genius way.